Hi, you guys. I just thought I'd make a quick video so that you guys can touch base and look at the study guide before the exam tomorrow. So, a couple questions that people have asked. Number one, do you need to know the names of people that did certain experiments? For the genetics of bacteria, yes, you kind of do. There's the Hershey-Chase experiment, Meselson and Stahl. There's the Davis experiment with the Davis U-tube, remember? And so you should know those names. And the reason why you need to know those names is because as you go on through biology, you're going to keep running into those experiments and those names. So it's a good thing to know. So yeah, who are the researchers that characterized each mode of genetic transfer? Who were the people that were responsible for showing conjugation, right? Transformation and so forth. The difference between auxotrophs and prototrophs. As it turns out, think about humans for a minute. In humans, we have the essential amino acids that we need to eat so that our bodies can make proteins. And those essential amino acids are the B vitamins that you get from eating meat, and usually red meat, right? So it turns out that bacteria are the same way. Some bacteria are not able to make their own amino acids. So what ends up happening in that particular case is you would have, let's say, an amino acid in a bacteria, and the bacteria changes it somehow so that now it's a different amino acid. Let's say you had a lysine and the amino acid adds something to it to make it a valine, okay? So in that particular case, you're looking at something that's being converted from an auxotroph to a prototroph. Prototrophs make everything they need to survive. Auxotrophs are missing things. When you put the two together and allow conjugation to happen, you can turn an auxotroph into a prototroph. And so that's what you need to know about auxotroph versus prototroph. Who is able to survive on their own on minimal media? Remember that the minimal media does not have all the essential amino acids. The organism has to make it themselves. Okay, can an auxotroph live on minimum medium, or do you have to have a prototroph? All right. Other than that, I would definitely make sure you know the difference between lytic and lysogenic cycles in terms of phage, but there's a lytic and lysogenic cycle for viruses as well. And then the other thing, all right, so that was one thing. The other thing that's going to be really important is what is a recombination frequency? What does it mean on a genetic map to have a recombination frequency? So remember on Friday we drew out two lines to identify two separate chromosomes, right? And then we mapped out A, B, C, and D. Remember we did this? That thing right there. You should be able to determine how genes line up on chromosomes. And so what was the rule? What was the frequency that meant that a gene was on another chromosome? Keep that in mind, all right? So I can guarantee you that that's going to show up. If you have any questions, please send me an email. Let me know what's going on. Other than that, number one, if you haven't started studying yet, at least go through and identify and answer the first 3 or 4 questions in each chapter. At least you'll get a little bit of coverage, okay? Number two, don't cram tonight. If you don't know it now, you might be able to absorb quite a bit in a few hours, but don't stay up till 4 or 5 a.m. It's not worth it. You'll be too exhausted by the time you show up. Get a good night's sleep, right? Maybe 6 hours. I wouldn't go less than 6 hours. Don't get too caffeinated because then you're just going to feel like throwing up. Eat a good meal. Come to school ready. Bring a Scantron. And let me know if you have any questions. All right, guys? See you tomorrow morning. Bye-bye.